Hello, my name is Peter Koenig. I am going to be talking about cardiac physiology along with my colleagues, Drs. Patel and Sen, and this is going to be part one. So I am going to be talking about some cardiac equations. I call these the equations of life and the circulatory system. The overall goals of these two lectures are to review basic concepts in cardiac function, determinants of cardiac output, review of the neonatal myocardium, and correlate these concepts with clinical and laboratory findings using pressure volume loops and clinical scenarios. What's not going to be covered are derangements of QPQS, i.e. right to left to right, and mixing lesions. However, in preparation for this, QP and QS are going to be introduced as it becomes part of all the following lectures. Gradient stenosis, regurgitation in relation to murmurs is not going to be covered. Neither is pulmonary hypertension. The fetal circulation, transitional circulation, and postnatal circulation in general, or neonatal shock. These are going to be given in other talks. The goals of this particular lecture are to review basic concepts in cardiac function and determinants of cardiac output. So we're going to start with equations of life, a review of physiology basics. A lot of this you should have learned in medical school, maybe put it away during residency, but this is time to revive it during fellowship and relearn it in more depth as you become more experienced. It'll make more sense, and I find myself constantly relearning things. So before we see the equations, you have to realize that the history, exam, and lab findings can give us the big picture and allow us to detect the derangement. And so just think about that. Each bit in the history, exam, and lab gives a little bit of a piece of the puzzle. All the equations that follow interrelate. So in general, a caveat is later on, you're going to consider all these equations to form a complete understanding of the cardiac function or dysfunction. So the big picture. The heart is a complex pump, and there's complex plumbing. The right heart only pumps to the lungs, and the left heart distributes blood to the body. Each ventricle has an atrium which receives blood and primes the ventricles. The circulatory system is set up in a way in which the brain and the autonomic nervous system and portion of the endocrine system with other organs control vascular tone and mean arterial pressure. Blood is routed where it is needed. There's also local control vasculature, or local vasodilation. So it's sort of involvement all the systems, the right heart, the left heart, the circulatory system. Remember, the brain is the chef de village. It controls everything, and it wants to keep itself alive, and then the other organ systems each contribute. So equation number one. Mean arterial pressure, MAP, is the diastolic pressure plus 1/3 the pulse pressure. And the pulse pressure, as you remember, is systolic minus diastolic pressure. So with a low systolic blood pressure, there's not a good way to achieve a good mean blood pressure. We'll have some questions and exercises later on to show this. If the systolic blood pressure is okay, you can probably get a reasonable mean arterial pressure. Your brain can control the systolic blood pressure more than the diastolic blood pressure, sort of this constriction thing versus local vasodilation. And then this all makes sense. If the brain is the traffic cop, it has to do major, with the autonomic nervous system, you're controlling major intersections, and the side streets are controlled with something local, and that's really what you want to have happen. And this will make more sense as we go along throughout all these lectures. Next one, equation number two, and questions will follow in the exercises. Blood pressure equals flow times resistance. You have to remember this. This comes back again and again when we do cardiac physiology and pathophysiology. If blood pressure were held constant, flow is increased and resistance decreases. Think about that. Think about this equation. If the blood pressure were held constant and resistance increased, flow decreases. If flow is held constant and resistance increases, pressure increases. And of course, if resistance decreases, the pressure decreases. If the resistance is held constant and flow increases, pressure increases. And if flow decreases, pressure decreases. We're just thinking about this equation, and we think about it again and again when you will apply it clinically. Equation number 3. Cardiac output equals stroke volume times the heart rate. So cardiac output correlates with flow in the prior equation. Remember I said flow times resistance? Well, that flow is really cardiac output. There's 3 things that govern stroke volume, provided the heart rate is not affecting any of them, which introduces a concept of minimum filling time. In other words, if they're very high heart rates, you may not be able to get a preload. So the 3 things that we have are preload, afterload, and contractility, and these really will dictate stroke volume. So let's talk about these. Determinants of stroke volume. The quick explanation. Preload is the initial stretching of the cardiac myocytes, muscle cells, prior to contraction. It is related to ventricular filling. Afterload is the force or load against which the heart has to contract to eject the blood. As aortic and pulmonary pressures increase, the afterload increases on the left and right ventricles respectively. Afterload changes to adapt to the continually changing demands on the cardiovascular systems. Afterload is proportional to mean systolic blood pressure. Contractility is the intrinsic strength of the cardiac muscle, independent of preload, independent of afterload, but a change in preload will affect the force of contraction. So there's some interdependence. Let's go into these a little more. I've got to throw in a picture for everybody, just for fun. You can spend a little bit of time. You can stop the slide if you want. I'm going to move on. So preload. It's the stretching or lengthening of sarcomeres in the cardiac myocytes prior to contraction. It's an end diastole. As sarcomeres are stretched further, there's an increased stroke volume as the sarcomeres then contract to the baseline length. So just by stretching and relaxing, the more you stretch, the more the distance that you're going to cover. There's also a more forceful contraction, which may be due to improved actin-myosin cross-bridging versus perhaps a length-dependent channel becoming activated. So there may be something else, but just the sheer lengthening, and you lengthen more, you can get a bigger change when you return to baseline. Preload can be approximated by the volume of the ventricle. It can also be inferred from the end diastolic ventricular pressure. However, the latter is confounded by the presence of mitral stenosis or ventricular noncompliance. So preload can be, you can use pressure and diastolic ventricular pressure as a surrogate of preload, but only if you don't have any confounding factors. Something to remember. So let's talk about reduced preload. If cardiac output is to be held constant and preload is reduced, other than increased heart rate, a reduced afterload or increased contractility can maintain stroke volume. So again, if you make some changes, you can have compensatory changes and still maintain cardiac output. So if you have reduced preload, some exam and laboratory correlates of this being reduced. Specifically, 1, you can have an increased heart rate and other signs of compensation, such as vasoconstriction. These are big things. And then you'll have signs of hypovolemia, specifically historical, clinical, or laboratory findings. History may have blood loss or diarrhea. On exam, you may have dry mucous membranes. You may have no urine in the bladder. You may have decreased skin turgor. You may have the lack of a large liver. In laboratory findings, maybe you'll have an elevated BUN creatinine ratio. How about elevated preload? Well, it may be needed, and it's physiologic. So if you're running, you're going to elevate your preload. So if one has an increased preload for an instance, or just an instant, the cardiac output will increase until reaching a steady state, and whatever initiated that increased preload is no longer a problem. So let's say if I gave you a fluid bolus, so increased your preload, but you don't need the preload, you're going to pee it off. So it's gone. So you get it, and then you get back to steady state. But if you actually need the preload, like if you're running, You can also have an increased stroke volume and the heart rate leading increased venous return. But you need that. You need an increased cardiac output, and that'll keep on going until you stop running. Does that all make sense? Let's go back. In general, if one has hepatomegaly or edema, it's a sign that preload is likely adequate or more, it's more than adequate, it's elevated, unless there's a discrepancy between intravascular and extravascular volume. And so in general, the concept, if you have an elevated preload, it's either needed and it's physiologic or it's pathologic. And you're going to know just by some simple things which one it is. Afterload. It's the pressure that the heart must work against to eject blood during systole, ventricular contraction. Can also be described as a pressure that the chambers of the heart must generate to eject blood from the heart, and thus is a consequence of aortic pressure for the left ventricle and pulmonic pressure or pulmonary arterial pressure for the right ventricle. The pressure in the ventricles must be greater than the systemic or pulmonary pressures respectively to open the aortic and pulmonic valves. As the afterload increases, cardiac output decreases, or stroke volume will decrease. Following Laplace's law, the tension upon the muscle fibers in the heart is the pressure within the ventricle multiplied by the volume within the ventricle divided by the wall thickness. This ratio is the other factor in the setting of afterload. And it's really, this is sort of the physiologic sort of equation of what is afterload. So if you compare a normal heart to a heart with a dilated ventricle, if the aortic pressure is the same in both hearts, the dilated heart must create a greater tension to overcome the same aortic pressure to eject the blood because it has a larger internal radius and volume. Thus, the dilated heart has a greater total load tension on the myocytes. It has a higher afterload. This is also true in eccentric hypertrophy, consequent to high-intensity aerobic training. It's also in concentric hypertrophy, you can have a lower afterload for a given aortic pressure. When contractility becomes impaired and the ventricle dilates, the afterload rises and limits output. This may start a vicious cycle in which cardiac output is reduced as oxygen requirements are increased. So think about this for a little while. Just let's go look at the equation. Here's Laplace's law, and it's calculated using this equation, the Young Laplace equation. The wall stress is proportional to ventricular pressure, or ADP, multiplied by the radius, or the end diastolic radius. It's really the volume. So you're thinking pressure and volume, or how big the ventricle is, divided by the wall thickness. So ADP is the end diastolic pressure in the left ventricle, which is typically approximated by taking pulmonary artery wedge pressure. End diastolic radius is at the end point, the midpoint of the left ventricle. H is the mean thickness of the left ventricle wall. Both radius and mean thickness of the left ventricle may be measured by echo. So think of what echo can do. It can give you a diameter of the left ventricle, can give you an LV thickness, and from that you can figure out what's happening with wall stress. Things that affect afterload: elevated blood pressure. That could be systemic or pulmonary. So systemic is actually increasing the afterload on the LV, and the pulmonary pressure is increasing the afterload on the right ventricle. Aortic valve disease. You probably don't think about this, but it is. Just like blood pressure. So aortic stenosis, the left ventricle must overcome the pressure gradient caused by the stenotic valve, in addition to the blood pressure required to eject blood into the aorta. Due to the increased afterload, the ventricle has to work harder to accomplish its goal of ejecting blood into the aorta. Thus, in the long term, increased afterload due to stenosis results in hypertrophy of the left ventricle to account for the increased workload. And again, I'll talk about this in a second, but and then aortic regurgitation, it increases afterload as well. And think about that, because the resultant LV dilation occurs from the regurgitant volume. This leads to increased LV wall stress as per our equation. It does not really reduce afterload. You'd think it would, because you've got blood you're ejecting, but it's coming back in low pressure ventricle. But think regurgitation is during diastole. The diastolic pressure in the aorta falls increasing the pulse pressure. But the bottom line is in AR, you actually increase afterload because of the LV dilation. In mitral valve disease, mitral valve regurgitation, you decrease the afterload. In ventricular systole, the regurgent blood flows retrograde to a lower pressure chamber, as well as antegrade across the aortic valve. So that's what's happening. The LV is both shooting out to the high pressure aorta and the low pressure left atrium, and so you have reduced afterload. However, the resultant LV dilation may lead to an increase in wall stress, countering the lateral reduction in afterload. So a couple things are happening, and you have to figure out, are you actually increasing or decreasing afterload with mitral valve disease, specifically with regurgitation. So let's think of the responses to increased afterload. Acutely, the heart will dilate. An increased afterload leads to a reduced stroke volume, and you're going to see this in your pressure volume loops. Thus, there's going to be a compensatory increase in the heart rate, and the heart's going to dilate. Two things that are happening. And so similar to an increased preload, the latter will improve contractility and increase sarcomere length, both of which counteract the increase in afterload to restore stroke volume. So stroke volume would go down with increased afterload, and then these other things are going to counteract that going down. So hopefully in the short term acutely, you'll be back and you can still maintain your cardiac output. Over time, that the LV dilation from the afterload and the increased wall stress will lead to hypertrophy, and that will restore wall stress, because remember, thickness is on the bottom of that equation, and as your thickness increases, you're going to normalize your wall stress. Okay, so initially dilation will increase wall stress, and then you increase your thickness, and that will then balance the equation. How about response to reduced afterload? Acutely, you can see an increased stroke volume. Chronically, you could have a reduction in LV mass, so opposite what happens with increased afterload. An example is in left transposition, where the LV thins because the LV is pumping to the pulmonary artery, and the LV thins as the pulmonary vascular resistance decreases in the neonatal period. So this is important for us, because if you've got a really thin LV, eventually, and this actually will happen in detransposition as well. Eventually, if your left ventricle is pumping to your pulmonary artery, your LV thins, and then will it or will it not be able to become a systemic pump or not? So you don't want to wait too long. You want to keep that hypertrophy present. Contractility is the innate ability of muscle fibers to shorten when they are activated by a stimulus independent of preload and afterload. If everything remains constant and contractility increases, it'll make the ventricle to eject more blood, which in turn will increase stroke volume. It implies that if preload and afterload stay unchanged, raising contractility will directly increase stroke volume and vice versa. So this is just a property of the muscles. This is on a cell molecular level. You increase contractility and you're going to increase your stroke volume. The ability to produce changes in force during contraction result from incremental changes of binding between filaments of myosin and actin. The degree of binding depends on the concentration of calcium ions in the cell. So factors that cause an increase in contractility work by causing an increase in intracellular calcium during contraction. So there's a cell molecular basis to it. I think in the next lecture, you're going to hear more about that. So now we're going to get to the next equation. Equation number 4. Ejection fraction is the difference between end diastolic volume and end systolic volume divided by end diastolic volume. That's sort of a 3D of volume sort of way of looking at ejection fraction. You probably have heard of shortening fraction, and it's very similar to ejection fraction, but instead of changes in end diastolic volume and systolic volume, you're just doing end diastolic diameter and end systolic diameter. You're just doing just linear instead of volumetric. So the EDV minus the ESV, that's the stroke volume. So stroke volume is a component of cardiac output. Contractility is a component of stroke volume, as is preload and afterload. However, people confuse this. Contractility is not the same as ejection fraction, which cardiacs call function. We usually refer, when we say function, we're talking about ejection fraction, or the correlate is shortening fraction. But we know that contractility, it's not the same, but it's inferred. We put it all together, what's going on with contractility. We put it together with measurements, we put it together clinically, and we figure out what is contractility. A reduced ejection fraction could mean an acute increase in afterload. Doesn't mean contractility is decreased. In fact, you may have a lot of the contractility may be very great. It just can't overcome the increase in afterload. Similarly, a normal or increased ejection fraction could reflect significantly reduced afterload, for instance, vasodilation with an AVM, even though contractility may impair it. So someone, and we see it, we always think of it with mitral valve regurgitation. We reduce the afterload and it looks like the function is okay, but that's masked. The contractility may actually be significantly decreased, but we can't tell because the overall function looks normal because of reduced afterload. So think about that for a few minutes before you go to the next slide, which I'm going to go to. More equations and terms. QS is, we just refer to QS as a systemic blood flow. And it is I've got these wrong. It's the LV stroke volume times the heart rate, and QP is the pulmonary blood flow. It's the RV stroke volume times heart rate. And I think in the next rendition, I'm going to have to correct this slide. It's amazing how you don't always see everything. So if QP is less than QS, so the ratio QP-QS less than 1, it means there's a right to left shunt in cyanosis. Think about that. We're going to cover that in later lectures. If QP is greater than QS, or the ratio QP-QS more than 1, means there's a left to right shunt. If QP over QS is 1, means there's no shunt. Or you can have funny things going on if you have complex mixing lesions. Or the other case, you can have QP-QS equal 1, or who knows where they're at, if you have the pulmonic and systemic circulation not talking to one another. In other words, they're going in parallel and not in series. And think of what lesion could that occur in? Transposition. It's going to be later lecture. Even more equations, really important. The conservation of mass. Assuming no leaks or other routes of blood flow, volume 1 equals volume 2. So stroke volume times heart rate for one system, whatever you're talking about, equals stroke volume times heart rate in number 2. Flow equals 1, flow equals 2, etc., etc. And just remember this for later talks on obstruction, regurgitation, murmurs, gradients, etc., shunts. So with this in mind, QS is always accountable to the body's needs. And that's a sort of a conceptual number. What is your systemic cardiac output? And that your brain is going to adjust it to your body's needs. Okay. And your heart is supposed to compensate. And you need to have whatever QS is, that's to be maintained not in shock, that is your body's needs. And if you don't have QS at that time, technically, you're in shock. Your heart's not able to maintain whatever that QS is. The absolute number, of course, can vary because your needs vary. And then the other part, you need to know what goes out to the body has to come back. It has to be that way. If not, over the times, the lungs would have all the blood flow with no blood to the body if QP over QS is more than 1 with each heartbeat. Think about it. Or we'd slowly have no blood going to the lungs and all the blood in the body if QS is less than 1. Again, if those ratios change, there's something else that's going on, but this always has to maintain. What goes out to the body has to come back. And QPS and QS, when the ratios change, means there's something else going on within the heart. And we're going to talk about this in those things in later lectures. Finally, we're sort of going to talk about the heart in big terms. The heart is the pump of the circulatory system, or chambers. 2 atria, they receive the blood and prime the ventricles. Got 2 ventricles to pump the blood. The RV pumps exclusively the lungs. The LV pumps the body to allow other organs to do their thing. Then there's central regulation, the brain and the autonomic nervous system for vascular tone primarily. The kidneys are there for fluid regulation, although they're also governed under autonomic control. But the kidneys are obviously important for preload. And then there's also local regulation or local vasodilation. That's going to occur at the organ level. So I like to put up just the diagram of just at least to start thinking of the pump in the circulatory system. QS is the cardiac output that leaves the heart, and that's distributed via the circulatory system. There are a lot of branches. It's not just a single tube that's leaving the heart and returning to the heart. You've got branches. So this takes a little thought. I always have to think about this. You've got mean arterial pressure, and you've got central and local regulation. The pressure in the aorta, in the branch vessels, if you measure systolic blood pressure, it's about equal. You think we think that, well, okay, your aortic pressure is this. If you put cannulas there, aortic's here, subclavian's, systolic's about the same. Of course, you have to ask, well, subclavian artery is smaller than the aorta. Why isn't there a gradient? You're going from a big vessel to a smaller vessel. But of course, the subclavian artery doesn't have the same flow through it that the aorta does. The aorta is bigger diameter. Remember that mean arterial pressure, although it's going to be the same, about the same, it is flow times resistance. And you've got a smaller vessel, so intrinsically you're going to have higher resistance, and you also have decreased flow in just that little vessel. But that ends up that the pressures in all those central branches are about the same. And it's all under control, the brain. The brain is the gatekeeper. So it's going to control where the blood goes, and if it's going to shut down blood to certain organ systems as needed to maintain cardiac output primarily to the brain. And the heart is also, if you don't have a heart, it's not going to respond. That's going to be totally under local control. Your coronary arteries supplying blood flow. The brain doesn't control that. That's going to be local, because obviously if your heart fails, this whole thing falls apart. So your brain is going to squeeze vessels down. It's going to be on not the big vessel level, but on your smaller arterioles, and it's going to control your blood pressure and keep your mean arterial pressure up. And it's going to reroute the blood, so it goes to the brain, it goes to the heart, it goes where it needs to. So there's the traffic cop, and you've got local vasodilation. Just always keep that in mind, and we're going to do that again and again when you get in different lesions and we start talking about shunts, etc., where it's coming from. But so the role, and I think of it when I go to my vacation house, we've got a well. Every time we turn on the shower, we hear the pump go on. We increase, if you will, the contractility. Boom, boom, boom. The pump's going on. If we have more things going on, then I'm going to have the shower. I'm going to do something in the kitchen, and now I turn on the outside hose. That pump is going to work a lot harder. But it's going to have to maintain the pressure in the system. That's what it's doing. The heart will increase the heart rate. One thing just to increase the output, but it's also going to increase how hard it squeezes. So in my case, how much this pump is pumping the house, because you want to maintain that pressure head, so it can go wherever it needs to go, and then you locally control. You turn on and turn off the faucets. You're going to route the blood in different places, but you always have that pressure in the central system, and that's maintained. And so that's the importance of the pump and the circulatory system. And it should start. Hopefully, it'll make sense. And these are the basics you need to know before we go on to some of the other talks later on. So why do you need this blood pressure? What's going there? Well, it's not just blood. There's a purpose for it. You've got oxygen delivery, and you can calculate this. It's cardiac output multiplied by the oxygen carrying capacity. Again, it's going to be, which is the amount of oxygen that's carried on hemoglobin and what's dissolved in the blood. And then you can add on the concepts of hemoglobin, fetal hemoglobin, etc. I'm sure that's going to be another talk about oxygen carrying capacity, especially in the neonate. But there's more. It's not just oxygen what you need the circulatory system. You need to deliver fuel, glucose, and you need to get the blood to supply to the other organs for removal or metabolism of substrates. So there's carbon dioxide. We need to remove it. We have to metabolize fat and protein, lipids, and we have fluid regulation. We have acid-base regulation. We have a complex system of carbon dioxide transport. So in terms of exercise, these are all the pulmonary circulation, the peripheral circulation, heart, the muscle, the lungs, they're all working like a set of gears, taking blood to deliver things and to get rid of stuff and to and get stuff back into the system. So carbon dioxide, oxygen, one we're getting rid of, one we're taking on. And so it's all distributed, and this heart's what's going to take us there, what's going to take it there. And then the brain's going to control a lot of things, especially if we increase our cardiac output. We have to figure out, well, where are we going to increase it to? So you have to incorporate other things. Are you eating? Do you need more cardiac output to the intestines? So you may have local vasodilation. And then when that happens, like turning on the faucet someplace, the heart may have to increase its cardiac output, and the brain's going to regulate it all to continue maintaining a normal mean arterial pressure, so it's there when it's ready to go when another organ system needs to go online. As long as there's no pathophysiologic derangements, this should all work, and it's all working like a well-tuned machine. And it changes, depends on now if you add things, exercise, I think cardiac output is going to be distributed in a different way to different organs. I hope that all makes sense. So the summary is the heart's a complex pump and a complex circulatory system. There's lots of regulators. I want to introduce all these basic equations and rules that describe the physiology. Memorize them, and you need to memorize these and just think about it. I know it seems really simple. You've done it before, but you're going to reapply these again and again when we start introducing complex heart lesions and derangements. And we're going to use the same equations again, believe me, and you're going to figure this whole thing out. It makes a lot of sense, and it's sort of fun. Okay, on to the next lecture. Thank you for listening.